Lidocaine: Synthesis and Function in the Human Body by Kamil Waszynski as part of an honors project at Michigan State University. In this video, I will be going over lidocaine and focusing on the molecule's origin, molecular structure, going over some properties, its application in the medical field of dentistry, function, and synthesis. Lidocaine was discovered by researcher Nils Lofgren and his assistant Bengt Lundqvist in 1943 at the Institute of Chemistry at Stockholm University. Lofgren and Lundqvist were experimenting with the molecule isogramine, which was first synthesized by Lofgren's chemistry professor and mentor. At the time, isogramine was known to numb the skin upon contact. However, it was never used as an anesthetic because it was outperformed by procaine. In experimental trials, a methyl group was added to the sixth position on the benzene ring of isogramine. They named this new molecule LL30 after their last names. Later, the name was changed to lidocaine. Pictured is an actual photograph of the original packaging in which lidocaine was distributed in the late 1940s. Lidocaine's full scientific name is 2-diethylamino N2,6-dimethylphenyl acetamide. Since it only contains carbons, hydrogens, nitrogens, and oxygen atoms, it is an organic molecule. The molecular formula is C14H22N2O and is composed of three important functional groups: an amide, a benzene ring, and an amine. The first functional group is the amide, and it is found in the middle of the molecule. It is made up of a nitrogen atom bound to a carbonyl and a hydrogen. At the bottom of the molecule, we see a benzene. This functional group is made of 6 carbons bonded in the shape of a ring. This benzene ring is aromatic because it follows Huckel's 4n+2 rule. The pattern of single and double bonds seen in the ring means it is conjugated, and therefore highly stable due to the overlapping of p orbitals. Lidocaine's benzene ring also contains 2 methyl groups at the 2 and 6 position. The last functional group found on lidocaine is a tertiary amine because it is bonded to 3 carbon substituents as seen in the picture. This amine is essential to lidocaine's function because it is the site of enzyme binding. The nitrogen bears a lone pair of electrons, which will be discussed in just a moment when talking about the molecule's solubility. Here are some physical properties of lidocaine. It has a molar mass of 234.34. Its melting point is at 68.5 degrees Celsius, and it boils at 181 degrees Celsius. Lidocaine solubility is 0.59 grams per liter of water. However, lidocaine is far more soluble in hydrochloric acid due to the amine functional group. The nitrogen has a lone pair of electrons, giving it a partial negative charge. When placed in the strong acid, HCl, the hydrogen ions dissociate and bear a partial positive charge. Dissociation between the nitrogen's partial negative charge and the H plus ion's partial positive charge results in protonation of the amine. Lidocaine is stored in hydrochloric acid for another reason, and this is to stop lidocaine from reacting with the oxygen in the air. Lidocaine plays an important role in the field of dentistry. Most dental procedures involve removing parts of the tooth and/or shaping the tooth to retain its proper structure. This is done in order to strengthen the tooth and for cosmetic purposes. Drilling is done using a handpiece and a tiny burr, which causes vibrations that can be uncomfortable and irritating to the patient. In the case of a root canal, the soft and sensitive tissue in the center of the tooth is removed. This is the tooth's nerve. Both of these common dental procedures can be very painful for the patient. Now, pain is really just an electrical signal that must be intercepted by the brain in order for the sensation to be felt. The pathway for these signals is via neurons, usually referred to as brain cells. In the human body, pain signals are transmitted by an extensive set of neurons called the dorsal root ganglion, found in the spinal cord. In the dental office, lidocaine is used as an anesthetic. This means it numbs parts of the body in order to reduce pain. It is a safe and an effective molecule. Lidocaine is packaged in small glass carpules with hydrochloric acid and epinephrine. As mentioned earlier, lidocaine is more soluble in the acid, and it helps prevent it from oxidizing in the air. The epinephrine constricts blood vessels so that the lidocaine isn't transported throughout the body through the bloodstream, which allows it to work for a longer duration. The carpule is inserted into a syringe and injected directly into the nerve of the jaw on the inside of the mouth where the procedure will take place. Lidocaine is a local anesthetic, meaning that it only affects the place that it is administered and not the whole body. The anesthetic properties of lidocaine are its purpose, but how does that really work? In order to comprehend this, we must first have a basic understanding of how neurons function. Neurons send and receive electric and chemical signals. Electric signals run through a neuron by the creation of an electric potential when sodium and potassium ions are separated. The separation of charge is accomplished by ion pumps which line the body of the neuron. In order to fire an electrical signal, sodium ions rush into the cell. Chemical signals, called neurotransmitters, are sent between connected neurons through a small gap of empty space referred to as the synapse. This is where the electric signal of a neuron is transformed into a chemical signal and transferred to the next neuron. The picture at the bottom of the slide illustrates this perfectly. A, B, and C are each individual neurons. The small space between them is the synapse, where the neurotransmitters flow. Lidocaine functions in the extracellular matrix, where it binds to proteins on the neuron's cell membrane due to its high binding affinity. This organic molecule targets three types of receptors: voltage-gated ion channels, ligand-gated ion channels, and G-protein coupled receptors. Both the voltage-gated and ligand-gated ion channels are responsible for moving sodium and potassium ions across the neuron's membrane to charge the cell. Lidocaine works by blocking these channels, resulting in a weak electrical gradient where the neuron cannot fire a signal. Without sending the signal to the brain, the body avoids the experience of pain. Lidocaine can also bind to G-protein coupled receptors called IGLURs. This GPCR typically releases the neurotransmitter glutamate into the synapse. Glutamate is the major excitatory neurotransmitter in the human body, which means that it promotes the firing of an electrical signal in the adjacent neuron. When lidocaine binds to IGLURs, it inhibits the release of glutamate. This results in no chemical signal to be sent between the neurons. This is important because even if the neuron manages to fire an electrical signal in the presence of lidocaine, the signal will not be able to be passed to the next neuron, and eventually the signal will die out, preventing it from reaching the brain. Lidocaine cannot be created by the human body, and therefore can only be synthesized in a lab. This is done in three main steps. Step 1 includes a reduction and an acid-base reaction. Step 2 has an addition and an elimination reaction. And step 3 is a substitution reaction. In step 1, dimethyl nitrobenzene is reduced by stannous chloride in an acidic mixture of HCl and acetic acid. This makes an ammonium salt via a reduction reaction. The ammonium salt is then reacted with potassium hydroxide in an acid-base reaction to make 2,6-dimethylaniline. This image depicts the arrow pushing mechanism of step 2 in the synthesis of lidocaine. 2,6-dimethylaniline is reacted with excess alpha chloroacetyl chloride in sodium acetate and acetic acid to form alpha chloro 2,6 dimethyl acetanilide. First, an addition reaction occurs between the 2,6-dimethylanilene and the alpha chloroacetyl chloride. Then, an elimination reaction occurs to deprotonate the amine group. Finally, excess diethylamine does a nucleophilic substitution on alpha chloro 2,6 dimethylacetanilide to yield lidocaine. Thank you for watching. I hope you learned something interesting about the wonderful organic molecule that is lidocaine. Also, thank you to my organic chemistry professor, Dr. Cooper. Without her, this project would not be possible.